Today we're going to talk about plant nutrition. I mentioned in an in an earlier lecture that plants are these unique creatures that have divided their organ system up into the organs that get energy and the organs that get nutrients. The shoots, the upper part of the plants, and the roots. They're different from human beings in this way. Today, now, I want to talk about how those roots actually get the nutrients out of the soil. Now, generally, plants get their nutrients through the root system by absorbing ions. Ions are those small charged particles. They're either charged positively, in which case they're called cations, or they're charged negatively, in which case they're called anions. And the charged particles come in through the root system, and I'll explain exactly how that happens in just a moment. Now, it's also the case that apparently some other kinds of molecules can be absorbed, very small proteins and things like that, but we're not going to really talk much about that. I think the general consensus is that most nutrient acquisition by plants is with ions, either cations or anions, principally either cations or anions. Now, the way that happens really is kind of a cyclic process. You begin with a plant that then dies, and then it begins decomposing. You get a decomposition process which produces soil organic matter. The soil organic matter continues decomposing until it breaks down into its ionic forms. And those ionic forms then are the things that are absorbed by the roots in a process that we'll talk about in just a moment. So it's a very nice cycle. Seems very sustainable and balanced, the balance of nature and all that kind of stuff. It's really cool. Die, decompose, recycle. Die, decompose, recycle, et cetera, et cetera. Now, the fly in the ointment is that the organisms that are involved in the decomposition process, well, they need ions too, don't they? They need to build their own bodies, and they have to use stuff to build their own bodies, so that many of these ions, they go into the decomposition process. That is, they're in the bodies of the bacteria and the fungi that are engaged in the decomposition process. So there's kind of a competition between the plant and the decomposition process, the decomposers. This is the bane of organic agriculturalists since time immemorial. If you put a bunch of leaves in your garden and you start growing your garden, what you find very quickly is that the garden doesn't grow very well. At least at the beginning it doesn't, because as the leaves begin decomposing, the decomposers that are decomposing those leaves, they're using the products of decomposition to build their own bodies. This, of course, is the logic of why we actually have composting, something that we'll talk about in a future lecture, not today. Okay, so there is this problem. Now, in addition to that problem, as far as the plant is concerned, the plant trying to get those ions out of the soil, the plant, if it's just a baby seedling plant and is growing very slowly, and if the bacteria have not built up their population to the point where they can take care of all the ions that are being produced by the decomposition process, why those ions, some of them just don't stay around very long. They leach out of the system. Now, when they leach out of the system, of course, they're not available to the plant. So the plant has this problem, especially when it's young. I mean, it can't really absorb all those nutrients. It's just beginning to grow, right? So a lot of the nutrients go to the competitors, the bacteria. A lot of it gets leached out of the soil, and so that is a huge problem, right? Now that problem actually is, I'm treating it as though it's an actual engineering problem. It's not. It just happens this way. That problem is actually solved by soil colloidal material. In the soil, you have small particles, and I'll tell you what they are in just a second, but you have small particles that are in the soil solution. Colloids mean that they don't settle out through gravity, that they sort of stay suspended in the solution. They're not in solution chemically, but they stay suspended in the solution. They're very, very tiny particles that stay suspended in the solution. So as you can see here, what happens is the ions actually get absorbed on the surface of these colloidal particles, and they go back and forth between the ion pool, that is the pool that can be taken up by the plant, and the colloidal structures, and the colloidal particles. So if you have a lot of colloidal particles, what that really means is that you have a lot of storage in the soil solution of whatever ions are adsorbed to the surface of those colloidal particles. and that becomes a little bit complicated, as we'll talk about right now. So you have this basic process here that I've just described. Now, in this lecture, I'm not going to talk about this whole process because parts of it are really fairly complicated. This whole bottom part of it right here, this is what we typically think of in when we talk, the subjects here we typically think of as soil science. So we'll leave that for another. There's going to be a couple of lectures on this part of this graph in a little later. But today we're going to talk about this part right here. This part is, we'll call this part plant nutrition. This is where the action takes place. Here you have a photomicrograph of a fine root, and you can see sticking out of the fine root are these little finger projections. These are actually evaginations of individual epidermal cells. These are plant hairs. Okay. Here you see it more diagrammatically, so you can actually see what it looks like. So in the epidermis of the fine root system, what you have are root hairs, and those root hairs are projections out of individual cells, and it's across the membrane of those root hairs that the actual absorption of nutrients actually takes place. Chemically speaking, it's a relatively complicated procedure, but from an ecological perspective, while we can understand it fairly simply, and we gain a lot of understanding of what particularities of particular nutrient dynamics are about by examining exactly how that happens. So that's what the root hairs are about. Now let's talk a little bit about those soil colloids that I mentioned that are so important because they actually represent kind of a governor in the situation, right? They represent the sort of a bank that the ions can be kind of trapped in until the plants are ready to use them. Okay, so these soil particles are, well, first of all, the soil is made up of particles, and typically we divide the particulate matter of the soil up into sand, silt, and clay, depending on the size of the actual particle. The relative proportions of sand, silt, and clay is a measurement that's taken for soils normally, and it's regarded as that's what soil texture is about, the relative proportion of those three category sizes of the particles. Now we're concerned in this lecture with the last particle size. We're concerned with the silt. The silt is very, very small particle size. The silt actually is the particle size that is the silt. Those are the particles that are the colloidal particles that exist in the soil solution. Okay, and they are of two forms, broadly speaking, of two forms. First of all, you have the structured clays. They tend to be crystalline structures that have a strong structure to them. Some of them don't. Some of them are fairly amorphic, and in a later lecture when we talk about soil science, we'll talk about the difference between highly structured and amorphic clays. But in addition to that, then you have the soil organic matter, which tend to be particles about the same size, more or less the same size. The one we're interested in are the same size. They're the colloidal ones. They tend to be amorphic also. They don't have a strong structure associated with them. This is sort of what they look like. Now, the important point of these colloidal structures is that on their surfaces they have electrical charges, as suggested, as indicated here. So knowing that the colloidal particles have electrical charges on their surface, you can begin seeing why the ions why the ions are connected with them, right? Because the ions are charged themselves, positive for cations and negative for anions, right? So if you have a lot of charges, what you're going to be, those charges are going to be attracting the anions and the cations, right? And as I indicated in the previous slide, when I showed you the picture, the bigger picture of the clay, of the structured clay, there were a lot of charges on it. There were supposed to be charges on it. That was that the diagram was supposed to represent. But most of them were negative, and that's kind of interesting. And certainly important for the dynamics of the way plants get their nutrients. But those particles typically have negative charges more than positive charges. They have positive charges also, okay? But they mainly have negative charges. And those negative charges, of course, then will be attractive to any of the cations that exist in the system. Now, here's a list of the most important, well, this is almost a complete list of the nutrients that we need to be concerned with when we're talking about agricultural production, okay? As you can see, there's both cations and anions. The ones that we're concerned with right now are the cations. And these four right here are probably the ones that are, I wouldn't say most important, but the ones that are most abundant in the soils, and they're the ones that are needed most abundantly as the plant is growing. You will note that there's two cations, calcium and potassium, and and three cations, calcium, potassium, and ammonium, and two anions, well, various anions associated with phosphorus, and then the nitrate is an anion also. Now, what you automatically can see, I don't need to tell you this, if those anions are in the solution, and you have the colloidal particles in the solution, well, the anions are going to be attracted to the colloidal particles, right? So a negative charge on one piece of clay, for example, is going to attract one cation to be absorbed onto it. So here I have an exploded view of one of those soil clay particles with all of its negative charges on top of it and positive charges. And you can see in red here, I've indicated I'm just using potassium and ammonium as the two cations that we'll be concerned with right now. But you have in the soil solution above that that colloidal particle, you have these ions floating around, and this is what you see. You see the ammonium attracted to the negative charges and the potassium attracted to the negative charges. Now, yes, they come out of the soil solution and they get absorbed on the surface of the colloidal property, but of course, their place is taken by other ions which diffuse in from the rest of the soil solution. Now, the way this happens is, and this is a really important point, is that things tend to go to equilibrium at a local level, at least. So if you could take, for example, let's suppose that little picture there is a little piece of the soil solution that you could have control over, and if you took all the ions and you made sure that they were in, well, let's say you put a piece of clay in there which charged, that clay will attract all those ions, as many as it could, the cations mainly, right? Well, then outside of that little isolated area, why you have a higher concentration of those very ions, and so they move into the space where the ions were before they were adsorbed to the surface of the clay. So in general, if you can imagine a piece of clay here, why that the anions and cations on the surface of that piece of clay will be in equilibrium. The proportions of potassium, ammonium, calcium, etc., will be in equilibrium with the proportions in the soil solution that is surrounding that colloidal particle, and that that will be a process that will continue operating that will continue operating always. There's a tendency to equilibrate. So now you have the basic information, which should make the next part of this, I think, obvious. Maybe not, but I think so. Here you have a diagram of a root hair extending out from an epidermal cell in the roots, and you have it extending into the soil solution. In that soil solution, you can see the colloids. You can see the clay colloid. You can see the soil organic matter colloid, and you can see all of the cations and anions that are swimming around in the solution there. So you can kind of imagine what forces are operating there. The cations and anions are being attracted to the surface of those colloidal particles, of course. Now, what the plant is doing, here I'll indicate here with 2 arrows, there you have an example of how the plant is actually pumping hydrogen out from the inside of the plant, pumping hydrogen out into the soil solution. Now, hydrogen is a cation, right? Hydrogen is positively charged. Now, what that means when that happens, if you remember that everything has to be in equilibrium, any colloidal particle near to the surface of that root hair, suddenly the positive hydrogen particles are coming out there, and they're going to compete with the potassium and the calcium and the ammonium, the other cations that are adsorbed to the surface of the colloidal particle, right? Which means that some of the nutrients, in this case we'll talk about calcium, potassium, and ammonium, they will be liberated from the surface of the colloidal particle because of the hydrogen that's taking the place on the particle. Now, the plant actively pumps the hydrogen out. There's other metabolic products that sort of diffuse out from the root system and that are also positively charged, but we're not going to talk too much about those. But you can see how you get this exchange of these cations, right? You get the exchange, you have hydrogen coming in and then the nutrients coming out. And then near the surface of the root hair, of course, well, you have an imbalance again. And what happens just sort of naturally diffuses into the system those cations that were liberated from the colloidal particles, okay? So you have 2 processes that are going on here, really. One is simply the process of diffusion. Ions diffuse into the root hair when they're at a higher concentration outside of the root hair than the cytoplasm inside of the root hair. But also there's an active operation going on here. There's active pumping of hydrogen out into the system, which creates the diffusion that happens. It's a little bit more complicated than this. I'm trying to give you a quick version of it to give you a sort of an impression and a kind of a feeling of how the whole thing operates. But that's the basic story. So with that basic story, you can easily see how the the ions get into the plant. All right, so to some extent they're pumped into the plant. To some extent they simply diffuse into the plant, depending on what the conditions are. Now, what happens when those conditions change? And unfortunately, ecology is that way. Conditions rarely stay constant, despite what people like to think of the balance of nature. Nature is always in the process of changing. What happens when the soil changes? Well, when you stop to think about just those colloidal particles, which I hope by now you see how important the colloidal particles are in all of the nutrient dynamics within the soil and the soil root system that we have. When you have changes, it's sort of contradictory, and initially it seems surprising how contradictory these things are, but think of what happens, for example, when you have a decay of organic matter. Well, everybody says that that's really good. Okay, you have the decay of organic matter. Organic matter decays, and that releases the nutrients into the ionic solution, so they're available for the plant to take up. But as those organic particles are breaking up, while they get smaller, they have fewer charges on them, right? So what's happening is you change the sort of storage properties of the organic material as the more organic material breaks down. So it's it's a good news and bad news as far as the plant is concerned. Now, this is a complicated process, and the whole issue of soil organic matter we'll talk about in a later lecture, because actually it's not easy to fully understand how organic matter functions in a soil, and it varies all over the world depending on what soil type we're involved with, and especially with what kind of agroecosystem we're trying to develop those particular soil types. So we'll leave that as it may. But one other thing associated with the changing environmental conditions that I would like to mention is that as the soil colloids, as the crystalline clays break down, well, what happens is the crystal structure almost always contains quite a lot of aluminum and iron. And so as the crystal structure breaks down, why that aluminum and iron, which may have been sort of trapped inside of the large crystal structure, well, as the structure breaks down, why those ions, iron and aluminum, they start being liberated into the soil solution. Now, iron and aluminum, first of all, the iron changes the pH quite a bit. It it lowers the pH so that you get more acidic kind of soils. And aluminum can be toxic, and you do sometimes find this aluminum toxicity in soils as they become older. So this is now a question of thinking about the age of the soil. As the colloidal materials break down, well, older soils, they will have broken down for a longer period of time than younger soils. As far as the soil organic matter is concerned, in more tropical situations where it's wetter and hotter for more of the year than it is in temperate situations, while you'll tend to have your organic matter breaking down faster, etc. You can imagine that those things are going to happen, and as in all generalizations, there are going to be exceptions to this, but you can get the basic idea of how that's going to happen and how it's going to be different depending on what the climatic conditions are where you're making these observations. So, for example, here you see this is the diagram I showed you before, and then here is a diagram supposedly of maybe that same soil, but after a long period of weathering. As soils get formed and then they weather. Okay, as they weathered, one of the things that happens when the soil weathers is you break down the clay fractions so that the crystalline clays tend to break down, and of course the soil organic matter, whatever was there in a long-term basis, is breaking down also. And so what you see here, you can basically get the idea. You see what you have here is you have all those aluminum ions there, you have iron ions there, and you have a change in the pH, which sort of interferes with the baseline pH gradient that existed from the surface of the root hair off into the soil solution. Because now into the soil solution, which used to have a higher pH because the hydrogen ions were not so common out there. Now, partially because of the iron and also the aluminum that are out there, why the pH gradient isn't as large as it was in the younger soils. And so you can imagine now, you can think yourself of what that has to do with how those cations are going to be exchanged from the soil colloidal surface onto the root hair. So this is that process that's so terribly important when you're talking about developing sustainable agricultural systems in the soil. One of the processes that's super important is this idea of the cation exchange capacity. That is, to what extent is the soil set up so that these cations are on the soil, on the colloidal surfaces, and so that they can be exchanged near the surface of the root hairs, and thereby give up the cations and sometimes anions that they actually have stored in that situation. So that cation exchange capacity, CEC, cation exchange capacity, extremely important aspect of the chemical dynamics of the soil system. Now we must consider this all-important topic that is now understood to be extremely important, not just in agriculture, but in ecology, in terrestrial ecology in general. That is, the majority of plants in the world, the majority plant species in the world, have associations in their roots with fungi. These are called mycorrhizal fungi. There are there's different types of mycorrhizal fungi. For all practical purposes, there are two major kinds. There's those that are called ectotrophic mycorrhizae, and that's an example. An example is here in the upper picture. And then there's our vesicular arbuscular mycorrhizae, and these are endotrophic in the sense that they're within the cell. So your ectotrophic are mycelia. There's a mycelial mat that covers the root hair itself, as you see in the picture, whereas in the vesicular arbuscular mycorrhizae, they're inside of the cell. They have these organs called vesicles. When they, let's see, what are they called? They're vesicles, but they look like little trees. Maybe you can see that in this slide right here, but that's why they're called vesicular arbuscular mycorrhizae. In both cases, what you have is the mycorrhizae, the fungi are infecting the roots of the plant, but frequently they infect the roots of the plants in a very mutualistic way, in a very positive way. And I say frequently because it's not always the case. And I'll explain that in just a second. But imagine what's happening here now. You have the fungus, the fungal body is inside of the cell in the root, and then the mycelia of the fungus, they extend out. And very frequently, the mycelia of the fungi extend way further into the soil than the roots themselves do. So if nothing else, and there are other things associated with it, but I'm just going to emphasize the fact that what the plant is doing, the plant is effectively foraging a much bigger area in the soil by using those mycorrhizae to absorb nutrients out of the soil. Now, there's quite a bit of interesting recent work that's been done on mycorrhizae showing that the conditions of the environment may stipulate whether the mycorrhizae are having a positive effect or even a negative effect. I mean, when you stop to think about it, you just look by looking at that lower picture there, you got a fungus that's infecting the cells of your body, you the plant, okay? Well, that maybe isn't such a good idea. Now, what we do know happens, frequently what happens is that fungus actually transfers nutrients from the soil into the plant. I mean, the plant basically gives it carbon and it forages for nutrients for the plant. However, under certain circumstances, if the plants are not nutrient limited, for example, why the fungus still can be there, still extracting carbon from the plant, but not really adding any benefit to the plant. So depending on conditions, you can have a mycorrhizal association with a plant which is either negative or positive. Now, in agricultural circumstances, we normally think of the mycorrhizae as being mainly beneficial, mainly adding to the absorption of nutrients in the system, into the plant itself. Now, that might not be true, and further research might provide us with more clues about that. It's an active area of research right now. But for the most part, as a rule of thumb, I think most people agree that the mycorrhizal associations in agricultural plants, at least, is usually a positive thing, that the mycorrhizae do aid in plant nutrition. Finally, getting back to the original diagram, where we see the overall cycle of how nutrients are cycled into and out of the plant and back into the soil, I hope I've convinced you that the process is, although it's a little bit complicated, it actually makes a lot of intuitive sense if you just think of the sort of the underlying chemistry and plant physiology involved in the whole thing. It's not that it's simple, it is complex, no question about it, but you can kind of see, I think, just from understanding a little bit about the structure, especially the details of this particular diagram right here, I think you can kind of intuitively see how plant nutrition is important for the development of agroecosystems.